Okay, I think we can start now. So welcome again to our Milner online seminar series. So today we have Lifebit, and particularly we have a team of Lifebit. Like I'm very thankful and really welcome all the speakers, Pablo, the CTO, and also the co-founder of Lifebit, and also with Thorben. I think he's a CBDO of Lifebit. And also Chiara, the precision medicine and human genetics lead at Lifebit. So, I mean, those three fantastic speakers will share the kind of next hour, and then I think they will tell us like an exciting story. So it's all over to you, Thorben. I think you're gonna organize those sessions. So yeah, over to you.
Absolutely, and thank you, Namshek, and thank you to everybody from the Milner Institute. It's a pleasure to be here, and thanks everybody for taking your time live out of your afternoon. I know some other people might be visiting or revisiting the session in their own time, and that's obviously great. So please do go ahead and share with those colleagues of yours who can't make it. And it's great to see such a fantastic outcome to discuss today. Really, the power and the potential that clinico-genomic data, so large cohorts of patients' data with the genomic data, whole genome sequencing, whole exome sequencing with clinical information, what that power can really bring out when it is connected. And connecting, you know, it's a simple word to say, but what does it really mean? And connecting, we'll showcase today how, especially here in the UK, a lot of advances are being made is to the federation between such trusted research environments, so secure places to analyze data. So we'll be diving into some of those topics today. You're obviously more than welcome to ask any kind of questions during the chat. We will be going through the presentation first, and then maybe stop the slide so you can see our faces and we can interact with one another. Obviously, you can also ask the questions live afterwards. So with that, we'll jump straight in. As Namshek already introduced us kindly. So I'm looking after the kind of the strategic partner side here at Lifebit as the Chief Business Development Officer, and Pablo representing our technology side, and he'll be diving in a lot of the kind of the technology questions behind what the topic today. So he's the CTO of Lifebit, and Chiara Bacchelli was actually a former associate professor at UCL and an active clinical researcher at Great Ormond Street Hospital. And here at Lifebit, she's heading the precision medicine division and is our human genetics lead. So I'm very pleased that both of them are joining me here to give the scientific as well as the technology experience and weight to the things that I'll be guiding you through. And it's going to be a bit of a back and forth. And of course, in the end, we're hoping for plenty of time for questions. And right, diving into the first one, data is obviously at the very core of that. So, Chiara, I throw the question right over to you. So, why is data so important? Let's start us off easy.
Yeah, thank you, Thorben. Yes, well, nowadays we have a huge amount of data, right? Data, genomic data, multi-omics data, phenotypic data, health clinical imaging, just to name a few, right? Data is absolutely fundamental in translational research, as everybody knows, for biomarker discovery, for example, with the opportunity to identify molecular markers and signatures, for disease processes, progression, prognosis, and improve clinical outcomes in general. Drug repurposing is another one, to shorten the process, to compound development and shorten the length of clinical trial, with the use of large integrated data set to facilitate screening of drug candidate. Patient stratification as well, whereby patients are stratified based on certain characteristics, including their genomic profile, to better target medication and treatment to a particular group of patients. And more generally, precision medicine, which really relies heavily on the availability of health data combined with genomic data and other type of data, and really aims at diagnosis, prevention, and treatment strategies, accounting for individual heterogeneity of the different group of patients. All of these need data. So just to give you an example of the impact that data has, so data really saves lives, as we normally say, right? Because these are a few examples. So for example, 5 times more rare disease patients get an accurate diagnosis by having a larger cohort of patients available in studies like large genomic studies like Genomics England, for example. 25% of these patients with the diagnosis, especially in rare diseases, were reported by the clinician themselves to be of immediate clinical actionability, as reported by the Genomics England paper that came out at the end of last year. In terms of regulatory approval, drugs with genetic evidence attached to it are twice likely to be approved, and generally more likely to have successful phase 2 and 3 clinical trials. Again, in a large study published in Nature at the end of last year, in collaboration between University of Edinburgh and Genomics England, 5 genetic markers were identified in COVID severe critically ill patients by performing a very large genome-wide association study. Some of these associations led directly to potential therapeutics approaches, as reported by the authors of the paper. So this showed that really data can be used to diagnose faster, develop better and faster drugs, and really understand the disease mechanism faster and in a more comprehensive way.
So then, Chiara, it's already a wonderful world. There's data, and it saves lives already. So there are no problems to be solved. Or what is the problem that we are discussing today with a lot more time?
Yeah, well, you see, so the problem is that often we as a researcher really struggle to get enough data, right, to be statistically significant and powerful for our studies. So especially this is true for rare diseases and really a rare form of cancer, right, where already you have a small number of patients worldwide, but they're also scattered again in, I don't know, data is in different institutions, research institutions, in different genome studies across the world, right? Genomics England, others, and data is siloed. So this, I think, is been and has been and still is a huge problem. So, and yes, and there are actually studies that show that research insights grow by 100 times by increasing the pool of data available 10 times.
Well, that's quite a relationship. So obviously, I believe it's not a rule as such, but we've come across a number of these. So it really shows the value of connecting the data together. And of course, when we look at that, the good news is already, there's a lot of good news to start with, especially here in the UK, is that of course there are large amounts of data existing in the world, and especially large amounts of data custodians who really create very high quality cohorts of patients for different disease area or population studies as a whole. So these are universities, these are national precision medicine programs, these are healthcare providers, and these are also private biobanks. So that's, of course, kind of the good news. But what we observe in our work here is that the vast majority of this data thus far is quite inaccessible. Actually, I think the WHO estimates that some 97% of the world's healthcare data is actually inaccessible, or where it is accessible, more often than not, it is unusable. And that's obviously because it's different data standards, but more importantly, it is fragmented across all of these institutions, and it is siloed. And the real reason for this siloing is, of course, the sensitivity, right? So we're not just talking, I don't know, telecoms users, behavior data. We're talking about people's whole DNA and medical records. So arguably the most sensitive data there is about a person. And at the same time, that data is very, very large. So, next generation sequencing, which has been with us now for quite some time, we can look at about up to 300 gigabyte for the raw data of just one patient. So you see that both the data volumes are out of control, and especially in Europe and led with efforts out of the UK, the data sensitivity is a very, very topical point. This patient data absolutely needs to be protected. And the regulator asks for this as well. And we see other parts of the world, like the Middle East or Asian countries, following on this very tight regulation. So kind of the former approach to the whole data science in this was we centralize it somewhere. You want to collaborate, great. You put it all in one pool, you analyze it there, right? You want to analyze data, you ask for data to be sent to you, or, it involves movement of this data, and, both the size as well as the sensitivity and the regulation just mean that's no longer feasible. And often people say, yeah, but it's anonymized, right? But the problem is this data is so specific. There's no data that could be more specific to a person than clinical data, and in particular DNA data. So the re-identification risk of this data is incredibly high. So basically already showing that just taking out kind of the name or maybe date of birth is not sufficient. You need to go through a lot more efforts to secure it. So that is understood, meaning that many of these data sets now are in silos. They're very protected, and that's great, but they're not working with one another, and this data is very fragmented. And now the solution to this, and the title of today's presentation is obviously already given it away, is the concept of federated analysis. So what does that mean? Easily, it means bringing computation and analysis to where the data sits instead of moving the data around. So we can call this in situ analysis. And that actually achieves a number of things. So first of all, the data custodian, or data controller under GDPR that is tasked with safeguarding that data now achieves security by design, because the data stays in the university, in the hospital, or when we're talking Genomics England, national initiatives. And they're completely in control of this data. They can control what's happening, who has access to it, and what is performed over it. But we'll be talking about this, but it also offers further potential. It offers the potential of linking these data sets globally and enabling actually global collaboration. So to bring this to life a little bit, we actually jump into the use case of Genomics England. Most people on this call, no doubt, will know Genomics England. For those who don't, it's an agency owned by the Department of Health here in the UK, and it was tasked actually at the time by David Cameron after the loss of his son to a rare disease to create the 100,000 cohort of cancer and rare disease patients and their whole genomes, and research asset that has never been seen before, a moonshot project at the time. And this said, Genomics England has been around for the better part of a decade now, but COVID actually brought another acceleration to it, and that was about, of course, COVID testing the health as a population as a whole, and the UK government reacting to it in the research response by actually creating a consortia around Genomics England, the NHS, the genomic study consortia, and funding bodies, as well as technology leaders, to make an additional data set, a quite unique and unprecedented cohort for any one disease, available to researchers. And that is the 35,000 cohort of COVID patients. And these has been collected from, and Chiara mentioned the paper already, was collected from 20,000 patients who were admitted to an ICU around the country, so had a severe course of the disease, and 15,000 cases that were lucky to have a much milder course, such as myself or the former health secretary are actually part of that cohort. And so this data, in conjunction with the 100,000 cohort as controls, was to be made available to researchers from pharma and academia to help tackle what has kept us busy for the last 2 years now. And when we look at this, what was the struggle that maybe Genomics England had, and we observe around the world, actually any biobank has, and that is the one between security and usability. Obviously, this data, very sensitive, can't be negotiated, needs to be safeguarded, and Genomics England has always been pioneering this. Lovingly we call it the Fort Knox of genomics. Here and there it's been called a data prison. So but whatever you call it, it's definitely the most secure place for data in the world, for data of this type, and a lot of innovation has been created there, like the airlock process. But on the other side, it meant for researchers that it can be harder to access this data, and it's siloed, so it's a bit limiting in terms of which analysis can be run over that data, and the data researchers struggle to combine the data with their own data as well as external data. And that's where now federated technology comes in step number one, and that is by basically interacting with the Genomics England computing environment in a federated manner, meaning bringing the analysis and computation from researchers directly to the data from outside, and And that enables that researchers can now query the data set, all women over 35 with breast cancer and a BRCA1 mutation, build a cohort of interest, and then run any type of analysis over that, really get that flexibility that research at its leading edge needs. But to dive into it a little bit more, but what it really takes from a kind of technology side, because this sounds that it makes sense and it's easy, but what it really takes at the core, I'll hand over to Pablo to dive into a little bit what actually happens what is this in situ analysis?
Yes, thank you. Thank you very much, Thorben. So as you said, in order to enable researchers to actually obtain or to get into novel insights in one of these biobanks, or what we call a trusted research environment, a trusted research environment needs to actually ensure at the same time two primary elements that Thorben has already highlighted. The first one is to be able to actually maintain security and control over the data, which has to be paramount. And the second one, it's also at the same time, it's to extend the usability and actually maximize the researcher's ability to actually carry out any or their own research. Now, the difficulty is how that can actually be achieved. So to draw some very simple, actually parallel analogy, similarly to what we have from our own operating system perspective in our own devices, what this system or this trusted research environment needs to actually be able to do is to have this system that actually allows to underneath manage and basically operate the data, access to data in an organized and very structured manner, while still actually allowing the users, in this case, the researchers, to actually bring any kind of application, install their own applications and use them within the context of that system or that operating system, which in this case for us, it's a trusted research environment. Now, in the case of this trusted research environment, this system needs to be able to actually manage and focus, particularly importantly, in 5 primary aspects or areas. The first one is from the operating perspective is how to work and how to manage a secure environment. Now, that can be quite complex depending on the research organization, as data might be stored in very different actually environments. In some cases, it's going to be cloud infrastructure. In some other cases, it's going to be on premises. But each of those actually have different flavors, different providers, different underlying actually technology, right? So there is a wide range of options available where the data can actually be stored and used to run the researcher analysis. Now, the important key here for this operating system is that regardless of the underlying environment, this operating system has to be able to run the theory and has to be able to manage and abstract away any technical details from infrastructure and facilitate the resources to the researchers to actually make the best use out of it. And here particularly, we mean about not just managing and provisioning access to data, but also especially the compute resources, which is actually going to be critical here, right? Because any operation that has to be carried out within that operating system, within the trusted research environment, being that running a specific analysis, running a script, a pipeline, an interactive session, and even running a simple, more or less simple complex query, it actually requires the management of compute resources on that specific and within that trusted research environment. So that's one of the main and primary elements that has to be taken care of. The second one is the federated data management, because researchers are going to be also needing to be able to actually connect to more and more data in the future. And this is something that we are going to be ending up touching more later on with further discussion on the federation. But researchers inherently, they have to be able to actually link and access very diverse and varied sources of data. And one of the key elements is here is to actually provide the ability to connect those extra or external data sources on a secure manner so that it doesn't affect really the security and doesn't pose any threat within the system. But also, especially, being able to actually avoid copying and carrying over permanently external data into each of these trusted research environments. And this is critical because we know that that further and further, there is an ever-growing number in terms of size, especially of biomedical data sets that researchers actually require. Now, the third main aspect and area that this operating system needs to be able to actually manage and focus on is cohort, what we call cohort browsing. Researchers, first of all, have to be able to actually discover or really understand what is the data that is available within a theory, not just provide bulk access to data and just show them and try to understand where they can actually find the right data files, the right information, and try to make use of it, right? Once they are presented with the data available, they need to be able to actually browse and query through the data seamlessly. And the research study might actually be focused on one specific condition in cancer, for example, very simply. So having that ability to get access and visualize the data that is currently available to focus on the specific cancer type that they want to focus their research is actually critical. And cancer type is just one very simple example, but in reality, researchers need to be able to create and subset cohorts with a very specific criteria that can be made up of a large number of actually phenotypes and even genetic markers that can end up characterizing a group of individuals for the study. So this theory and that system needs to be able to provide straightforward manner access to that data catalog and access to that wide variety of characteristics before being able to actually run any analysis. Now, the fourth and follow-up actually element that the system has to be able to take care of is flexibility for running analysis, because researchers need to be able to carry out a wide variety of analysis in order to obtain their insights. And this can be really pretty much any possible analysis that anyone can think of. So the system has to be able to provide flexibility to bring any application and run any analysis that is going to be required, and not just giving access to a predefined list around a number of tools and libraries from different areas like genomics, transcriptomics, and imaging, and you name it. The most typical actually use case for, especially for example, by ourselves, bioinformaticians, is to have the perspective of being able to create and integrate and bring our own newly developed workflows and analysis into the current system. And having that flexibility for a researcher to allow them to bring without any restrictions, use them, validate them, and running them live is actually a critical need for researchers. And that also touches on the capability of not just being able to access data, but also being able to actually connect and even federate the locations where these applications might be actually stored, which is also key. And this might even also, of course, span to interactive type of analysis. So not just batch or different tools or libraries that our informatician will typically run, but also have the possibility of running iteratively and very swiftly interactive sessions on real time in the system that they can actually allow the researcher to have this very quick feedback loop to iterate, for example, over their own statistical analysis. And the fifth and the last main area that is quite key on a trusted research environment is the aspect of collaboration. Now, researchers, we know that always or typically they work in a collaborative manner with some of their peers at different levels. In some cases, they might need to be able to create, define cohorts and run analysis and work together in order to get to the satisfactory results. But in other cases, they might just need to, for example, share some of their analysis results so that a colleague can actually take it from there and run subsequent and follow-up analysis. And in some other cases, they might actually just require to provide read-only access to some of the results to their colleagues. So providing such a collaborative environment needs to be a critical aspect of the collaboration within the collaboration layer within the system. And it's something that sometimes we take for granted, but and it might be actually seeming obvious to us, but it's not always the case, and it's not always provided across biobanks and trusted research environments.
Thank you, Pablo. That's the perfect cue, because now we really understood what it means to analyze this in situ, but obviously why we're here today to discuss is then also how does this collaboration across different institutions work. And this is where basically the element of connected cohorts comes into play. where hang on, there's a sorry, there was a slight lag here, where now Genomics England as well as other biobanks are basically of sorts joining a network, if you will, of institutions that built on a federated architecture. So that is, as you know, from the US, institutions like the Jackson Laboratory, all the way to the Hong Kong Genome Project in Hong Kong, that are built in a way that data can now interact, right? And as we'll hear later today, this is even driven further from industry, the likes of Boehringer Ingelheim. But here I want to highlight especially the partnership actually between Genomics England and the NIHR Cambridge Biomedical Research Center at Cambridge University, so obviously very close to the Milner Institute, and what is happening there in the new Federation project is actually brings this to life. And for this, it's worthwhile looking at, you know, wonderful. We have TREs now in Genomics England, and that's a beautiful thing. So researchers can access this data in a wonderful way, in a secure way. They're very flexible in the use of over that data in situ. And that same researcher might though also be approved to access data that is held by the NIHR Cambridge Biomedical Research Center, a beautiful cohort there, and fully enabled there. But what's the problem? The problem is that between these two institutions, and this is just two examples, like the fact is between all institutions, there's still this divide, there's still this siloing, right? While data is very usable in place, those data sets do not yet go together. And that is something that we are very excited to work alongside Genomics England and the NIHR Cambridge BRC, as well as the Eastern HSN, in a consortium that together were awarded a grant under the DARE UK program, funded by HDA UK and ADR UK and the UKRI, to actually establish this communication layer between trusted research environments, so that federation across trusted research environments can be established. And of course, you know, I can say this very easy, but the devil is as always in the detail. And so I'll hand it over back to Pablo to dive into a little bit more of the architectural layers and also actually the different forms of federation that we have been touching on here.
Perfect. Thank you very much, Thorben. Again, yes, we are very much thrilled to be developing this first multi-party federation under the UK funded project, along with the consortium that Thorben just mentioned, bringing together Cambridge and AHR and Genomics England. And what this effectively brings together for the researchers is having the ability to really learn, to have access and to run much larger and more well-powered actually studies, and making the process of combining these data sets much simpler for any of the researchers that can have access to this multi-party federated approach. Right? This is obviously something that that focuses on the federated approach that we have been working on. And in this case, really, what federated approach should mean for the researchers, or from the researcher perspective, is the fact that we will jump from, as Thorben was saying, having two separated silos from Genomics England and NIHR, and to have actually a connection which is going to be bringing a single layer of visibility to a researcher that will give them access as if all the data was actually accessible and combined together as a single cohort, as opposed to having to be able to browsing and navigate through both cohorts or data sets separately, right? And in terms of to touch upon the federation approach, or the federated architecture, I'll briefly try to mention how that federation approach has been evolving across time. It's been something that there has been an evolution, and there are different use cases actually that have tried now to approach different problems and have used federation in order to further generate and gain insights into it. At the early beginning, just have to very clearly differentiate what is it, which might seem obvious again, but what is it that it's not federation at all, right? And in this case, you can just think about purely manual steps with obviously humans very much heavily involved, during the process. And humans in this case are going to be having to go through either Genomics England, NEHR, Cambridge Data, UK Biobank, and other data sets, and have to actually get access through a very manual process, get into the data, maybe get shipped a copy of the data itself, have it to move it somewhere, some maybe server cluster that has accessibility to these computer resources, and then run this analysis separately, right? And that becomes a very cumbersome process, not just for bureaucracy purposes, to get access to the data, but also in terms of how to bring the data together and try to combine it and make meaningful analysis out of it, right? And one clear aspect in here, no federation, is the centralized compute, obviously. There is one next step, obviously, before we get to the full federation, is what we call the partial federation. And this, what we are here highlighting in this architecture, is having federated access to the data. And what this really means is to be able to actually have distributed databases and data sets, which can then actually be used in order to analyze, at least from a compute centralized mode, but still being able to have direct access and links to this distributed data without really the need of copying over entirely and permanently the data, right? So this simplifies at least at the step, and it provides at least the capability to stream and just have direct read access to the data just in time for the analysis. Now, the final and full federation model that we are highlighting and displaying here, it provides a combination of federated data and also compute access, right? And what this really means is that we still have the distribution of the data, of the databases and the data sets, but at the same time, together with the data, we also have distributed access to compute resources. And what this really actually enables is that any query that has to be done in order to generate any analytics or to process any of these distributed data sets, it will happen on its dedicated resources that are living together with the data at the same time. And the same actually happens for any joint analysis that is going to be running across distributed data sets. Now, this full federation model tries to focus on especially strong privacy preserving analysis model for federated analysis and federated data sets. And this is also the case for genomic data sets. And the aim here is to really enable genomic data processing within and across organizations in order to run joint analysis, but without breaching any privacy or compliance aspect at the theory level. For this type of federated analysis, we have to define two main layers in order to deliver joint analysis or analytic insights. At the first layer is the analysis and compute that actually happens within each organization theory. Each theory can then be seen as its own state, which actually controls and owns and manages access to its own private data, as well as its own compute resources, which in the case of, for example, Genomics England, it might be a cohort data set of 100,000 individuals. Now, within that theory or federated state, granted the permission, genomics insights are actually generated, but solely at the theory data level. Now, in this case, the analytics are obviously subject to the rules and governance of that particular state, which means that all compute and data resources utilized will always belong to the theory and will stay and happen within the theory, period. Now, the same principle applies for any type of actually analytics, not just for any joint analysis that we might be speaking from bioinformatics or genomics perspective, but this can also apply to simple or complex queries in order to be able to find subsets of patients and samples that might match certain clinico-genomic criteria, which might help the researcher to actually stratify cohorts for their specific research and analysis. And any of these federated queries themselves are also going to be handled separately and subject to the access within each theory. Each query is going to be going through each of these theories and might actually require even the computation within theory on the fly of certain statistics that are going to be then returned and aggregated later across all the multiple theories before being able to actually present back to the researcher what's the cohort overall across theories and how does it actually look like, right? And this model of federation architecture has been used across many different areas. Actually, federated analytics is one particular example, which we are using on an everyday lives. We use it again in our own devices like mobile phones, and we use it for any kind of application that we can think of. This can be for maps, for just when I'm actually trying to travel or trying to commute, which I'm always getting lost, even with the help of this wonderful federated analytics. But this might be also related to my lifestyle, exercise that I'm doing, dieting, and so on, right? So all these actually applications on devices are constantly extracting and generating analytics that are sent over to somewhere else and aggregated all together, and then provide further insights to multiple users and society in general. And this is also the case actually for the full federation model in the case of federated machine learning, or more particularly the federated machine learning training actually steps. And the nuance actually here for the federated training approach is that when a machine learning training process is happening on this full federation process, what actually is going to be extracted and retrieved from these federated states is actually not going to be really data insights, but it's going to be really a local model, which have been generated within each of these states. Now, these particular models that are trained separately are part of these insights that then can be aggregated at the centralized compute layer, and then be able to redistribute, of course, to be able to proceed with the machine learning training process. Now, in this federated model, I mentioned before about the first layer. So obviously there is also a second layer required to generate this federated analytics for a researcher, for example. And the second layer is where the data safe haven actually comes into play. And the first layer that we mentioned before is the one that actually, once the analysis has been completed within each theory, it generates some aggregate level results, which always stay within the theories, right? Now, there has to happen an additional step in order to convert from single theory aggregate results to cross theories and federated analytics. And in order to do it so, we actually need this additional secure place or safe haven environment, which is created. It's outside each of these theories, and it will only have access to a limited aggregate tier level data from both of the theories. And we'll have the responsibility of further orchestrating and managing the generation of this cross-theory federated analytics, taking care of pulling the data from each theory, from the analytics, aggregate level analytics perspective, and also taking care of all the aggregation and compute resources that are required to get to that level. Now, in the end, after these federated analytics have been generated, the aggregate level results will have actually to go through, obviously, a strict airlock process in this case, in order to ensure that no sensitive data is going to escape the theories, which currently right now is going through a manual process. Now, additional work is going to be required in order to be able to automate the process and the policies used for an airlock, for airlock, which can be a bottleneck for researchers. Now, in the future, we expect to be able to automate the setup of granular permissions for data analytics requests, which will actually allow organizations to identify and approve beforehand which users are actually allowed to access data and for what purpose. So in this demonstration of multi-party federation, as Thorben mentioned before, we are going to be actually demonstrating and showcasing how to be able to allow researchers to analyze NIH shared data and GEL data jointly, while using a system that conforms and develops on top of best practices and also community and industry standards like GA4GH, and try to also help in terms of contributing additional best practices and open standards for multi-party theory federation.
Amazing, Pablo. Thanks for that deep dive. And of course, we could be talking for a long time about all of the intricacies of this. But Chiara, what are actually now some of the different kind of research approaches that are possible because of this multi-party federation?
Yeah, so if I just link him back to the Cambridge NIHR, for example, very briefly, as Pablo already mentioned, they will perform a joint parallel query over the GEL data and their own data set, and really leveraging the value of having in both cohorts normal and tumor whole genome sequencing samples for cancer patients, right? And between those two data sets combined, that will be the largest cohort analysis of whole genome sequencing cancer worldwide, which is amazing from the point of view of the research, obviously. So they're planning to perform, for example, as an example, joint germline analysis and somatic tumor analysis to really explore the interplay between predisposition variants and somatic mutational signature as well. And also aiming to use, obviously, all the distributed clinical information that comes with the genetic data to really understand the clinical impact of the analysis that they're trying to do.
That's fantastic, and that's obviously a very huge academic focus coming out of both Cambridge and Genomics England. But there's actually that big interest from industry in this whole field as well, where Federation plays a role, doesn't it, Chiara?
Yeah, absolutely, Thorben. Yes, for example, Boehringer Ingelheim, obviously is a big pharma. They have absolutely so many relationships with the different biobanks all over the world. They have a huge global network, right? But what they want to do is to try to analyze the data in those different biobanks jointly, right? So what they're doing at the moment is building a technologies or different technologies to really address unmet clinical needs by connecting those global biobanks through multi-party federation. And why are they doing that? Is obvious, but to accelerate translational medicine, accelerate the development of innovative medicine, really by integrating seamlessly world-leading population clinical genomic data and disease-focused cohort in a secure environment for analytics and research. So basically, they want allow the researcher, obviously, to gain a joint view of both the internal data that they have at BI together with those external biobanks, and query those data combined and jointly run analysis over it. All of this without local regulation or security being a hurdle for them through federation.
Thank you, Chiara. And I think this last example is a perfect close, where we really see an acceleration of this, federation word that has been used so much by it's been in the media now for a long time as well. Where especially here in the UK, we see a moving together of leading institutions with huge interest from other global institutions to join in on kind of this network character and that additional acceleration through global drug discovery and drug development companies such as Boehringer Ingelheim who really now want to grasp that value of connected data. And clearly we are steering in a world of connected data, and it's fantastic actually here out of the Golden Triangle, out of London, Cambridge, Oxford, and to help contribute to this with institutions such as Genomics England, the NHR BRC, and the other big cohorts that we have in this country, as well as then we're a British company, we're working to make this a reality. But right now I think you heard enough from us, so we want to pull it to a close and invite you guys to any questions. And of course, if anybody's shy today, you're more than welcome to also reach out to us. So we popped our email here, hello@Lifebit, pretty simple. So feel free to reach out to us.